Hello dear students, so today I am here with some multiple choice questions from developmental biology on your demand. So my first question is, the egg of a frog is centrolecithal, telolecithal, microlecithal and alecithal. Centrolecithal egg is actually found in insects, right? So how the centrolecithal egg is, I have already made a video on the type of egg, so there you can go and watch the full video for complete explanation with the diagrams. In centrolecithal eggs, the yolk is present in the center and cytoplasm is present only in the periphery as a thin layer, and a little bit of cytoplasm is also present around the nucleus as well, right? So centrolecithal egg is found in insects, and telolecithal egg is a egg in which the yolk is present in a moderate amount, and yolk is present only on the vegetal hemisphere, right? So if this is the vegetal hemisphere of the egg, yolk is present only in this hemisphere and cytoplasm is present in the animal hemisphere, right? So this type of egg is telolecithal and it is found in frog. Next, microlecithal egg means yolk is present in a very little amount and it is found in Hadmania and Brachiostroma. Next, alecithal egg means yolk is totally absent and it is found in mammals, right? In frog, the structure known as grey crescent is found in, options are A. fertilized egg, B. unfertilized egg, C. brain and D. eye. So first of all, let's understand what is grey crescent. So if this is the egg of the frog, and let's say if this is the point of entry of the sperm into the egg, then in the area just opposite the point of entry of the sperm, in this area, the pigments from the surface of the egg, they migrate into the interior, right, from this area, crescent shaped area. Since the pigment, it migrated from the surface into the interior, so this area, it becomes grey in color, right? So that is why it is known as grey crescent, right? So the question is whether it is found in fertilized egg or unfertilized egg. It is found in the fertilized egg because it is formed during fertilization when the sperm, it enters into the egg. So the correct answer is fertilized egg, A. So the next question is, the grey crescent of frog's embryo represents the future dorsal side of the embryo, ventral side of the embryo, anterior side of the embryo, posterior side of the embryo. So the area, correct answer is dorsal side of the embryo. So let me explain you how it is. So if this is the frog's egg, then as I told you, this is grey crescent, and this area, this side of the egg will become the dorsal side. Then the opposite end, or we can say the point of entry of the sperm, is known as this area will become the ventral side, and the animal hemisphere side will become the anterior side, and the vegetal hemisphere side will become the posterior side. So this is how the symmetry of the egg changes after the formation of the grey crescent or after fertilization. Initially the egg has radial symmetry, but now after fertilization it has bilateral symmetry, right? If two blastomeres of an embryo at two-celled stage are separated from each other, one will die, other will develop into a complete embryo. Each of them will develop into a complete embryo. They will die. They will give two half embryos, one developing only left half and the other only the right half. So we are talking about the two blastomeres which are formed from the zygote after first cleavage. So after first cleavage, the blastomeres which are formed, they are totipotent. Totipotent means they have the potential to develop into a complete embryo. So if you separate these two blastomeres at this 2-cell stage, then they will develop into complete embryo. That is, this will also develop into an embryo and this will also develop into complete embryo. So each of them will develop into a complete embryo. So the right answer is option number 2, B. Right? Importance of gastrulation in frog is: A option, invagination and convergence. B is involution and epiboly. C is invagination, involution and epiboly. And D is convergence, involution and ingression. So the correct answer here is C, that is invagination, involution and epiboly. So these are the different movements of the blastomeres during gastrulation. So what is invagination? Invagination, actually it involves the folding of the epithelium into the interior of the embryo, like this. So here the cells do not lose contact with the adjacent cells in the epithelium. So basically you can say it resembles like if you push a rubber ball with the thumb inside, if you push a thumb inside a rubber ball, then it shows invagination movement. So next is involution. So in involution, actually there is a the epithelium, it bends on itself, it bends on itself like this. So that is involution. So it bends on itself and then it spreads in the opposite direction, right? So this type of movement is involution. Next is epiboly. So in epiboly, the cells, they basically they spread over the other cells. Like if this is one layer of the cells, then these cells, they will spread along the perpendicular to the apicobasal axis, means they will become flattened. इस तरह से वो flattened हो जाएंगे. ये क्या है, इनकी apical surface है, and this is the basal surface, the lowermost surface. So they extend in the axis perpendicular to this apicobasal axis. They become more flattened, so actually they spread over the inner cells. Or we can say in case of frog, there are micromeres and macromeres. तो जो micromeres हैं, they spread over the macromeres by epiboly. Epiboly means वो flatten हो जाते हैं, and this is how they spread over the macromeres. Question is, during gastrulation, small groups of independent cells separate from a primary layer and migrate into the blastocele as a new layer by a process known as, first is convergence, second is ingression, third is involution, D is delamination. So first of all, what is convergence? Convergence में क्या होता है कि जो cells हैं, they extend along a single axis. एक सिंगल एक्सिस की तरफ उनकी ग्रोथ होती है. लेट्स से कि अगर ये सेल्स यहाँ पर ये 4 लेयर्स में है, राइट? सो दीज सेल्स, दे लूज कॉन्टैक्ट विद देयर नेबर्स एंड दे एक्सटेंड फॉरवर्ड अलोंग सिंगल एक्सिस. तो इस तरह से एक सिंगल डायरेक्शन में इनकी ग्रोथ हो जाती है. दिस मूवमेंट इज़ नोन एज कन्वर्जेंस. नेक्स्ट इज इनग्रेशन. सो इनग्रेशन में क्या होता है, let's say this is a layer of cells, right? So some of the cells, they change their shape and they lose contact with the neighboring cells. So let's say अगर ये cells हैं तो इनकी ये shape change हो गई यहाँ पर, right? And this is how. So these cells here, their shape has been changed. Now they will separate from their neighboring cells. तो अब ये क्या हो जाएगा, इनसे separate हो जाएंगे. तो ये layer तो ऐसे ही रहेगी और ये जो cells हैं, now they will migrate inwards into the into the embryo, to their proper place where they have to go. So this process is ingression. So here the cells, they change their shape and they migrate into the blastocele and they form a new layer. So this process is known as ingression, and this is the right answer. C, involution we have already discussed. Delamination, in delamination what happens that the cells they separate as a sheet, एक पूरी sheet जो है वो separate हो जाती है from the above lying layer of the cells, that is known as delamination, and mostly जो endoderm formation it takes place by delamination. So the answer for this question is ingression. The termination of gastrulation is indicated by, कब पता चलेगा हमें कि gastrulation has terminated, it has finished, right? Gastrulation complete हो चुकी है by closure of the blastopore, closure of neural tube, obliteration of archintron, obliteration of blastocele, right? क्लोजर ऑफ ब्लास्टोपोर क्या होता है, जब गैस्ट्रुलेशन होता है तो एक कैविटी बनती है जिसको बोलते हैं हम आर्केंट्रॉन, right? So the opening of the archintron is known as blastopore. So closure of the blastopore is actually not the last step of the gastrulation. Next is closure of neural tube. No. Obliteration of archintron. Obliteration means no, archintron is actually formed during gastrulation, so there is no obliteration of archintron. D option is obliteration of blastocele. Yes. How? गैस्ट्रुलेशन में क्या होता है, जैसे जैसे सेल मूवमेंट्स होती हैं. व्हाट इज़ गैस्ट्रुलेशन? गैस्ट्रुलेशन इज़ एक्चुअली द प्रोसेस विच इन्वॉल्व्स द मूवमेंट ऑफ द ब्लास्टोमियर्स to their respective position so as to form 3 germ layers, right? That is ectoderm, mesoderm and endoderm. So as during the process of gastrulation new cavity is generated which is known as archintron. So as the archintron is formed during gastrulation, so blastocele it reduces in size. So obliteration of blastocele is the right answer. Amphibian embryo is anterior lip of blastopore, posterior lip of blastopore, dorsal lip of blastopore, ventral lip of blastopore. So the correct answer is dorsal lip of blastopore, right? So dorsal lip of blastopore, why it is known as primary organizer? Because this dorsal lip tissue it organizes the anterior posterior axis of the embryo, क्योंकि ये anterior posterior axis को organize करता है. So that is why it is known as the primary organizer. For more details you can watch my video on organizers and induction. Regeneration of salamander limbs, options are, is an example of epimorphosis. What is epimorphosis? Where a lost organ of the body can be replaced by proliferating new cells from the surface of the injured part. तो injured part पे नए cells बन जाते हैं and they it leads to the regeneration of that lost organ. That is epimorphosis. So this is correct. Second is it involves the formation of apical ectodermal cap from the epidermis. Yes, from the epidermis जहाँ पे wound होता है वहाँ पे जो epidermal cells हैं they divide to form an apical ectodermal cap. This is true, and it also involves de-differentiation of the cells into a blastema. So वो जो cells they undergo de-differentiation. Yes, it is true, they undergo de-differentiation. Third point is retinoic acid plays an important role both in de-differentiation of cells to form regeneration blastema and in re-specification processes as cells re-differentiate. This is also true because in regeneration actually what happens, in epimorphosis the cells first they undergo de-differentiation, they divide to heal the wound and to replace that lost organ, and again they re-differentiate into that particular organ. So all the above. So the correct answer is option D.